Bismillahirrahmanirrahim. In the previous lecture, we have discussed the catabolism of purine nucleotides and the related disorders, that is the hyperuricemia. Another disorder that is related to the purine nucleotide metabolism is the hypouricemia. This hypouricemia is due to the deficiency of adenosine deaminase. As we know that adenosine deaminase catalyzes the deamination of adenosine to convert it into inosine. Thus, with the deficiency of adenosine deaminase, there is accumulation of adenosine. Then, after the accumulation, this adenosine is converted to its ribonucleotide or the deoxyribonucleotide forms by the cellular kinases. Thus, as the deoxy ATP level rises, it inhibits the enzyme ribonucleotide reductase. This ribonucleotide reductase converts the ribose to the deoxyribose form. Thus, with the inhibition of ribonucleotide reductase, the production of all the nucleotides which contain deoxy form of ribose become deficient. Thus, due to the deficiency of nucleotides having deoxyribose, results in the failure to synthesize DNA, and then leads to the failure of the cell division. The DATP and adenosine that accumulates due to the deficiency of adenosine deaminase, it leads to the developmental arrest and the dysfunction of lymphocytes. Thus, the adenosine deaminase deficiency causes the severe combined immunodeficiency disease, which involves both the T and B lymphocytes. Without appropriate treatment, the adenosine deaminase deficient child usually dies before 2 years of age. Another cause of hypouricemia is the deficiency of xanthine oxidase. As we know that xanthine oxidase catalyzes the oxidation of hypoxanthine and xanthine to the uric acid. Thus, with the deficiency of xanthine oxidase, the formation of uric acid is inhibited, and then there is accumulation of hypoxanthine and xanthine. Thus, these both purine bases then excreted in the urine. So the concentration of hypoxanthine and xanthine is raised in a patient with the xanthine oxidase deficiency. This accumulation of hypoxanthine and xanthine may also lead to the stone formation in the kidneys. Now we will discuss the synthesis of pyrimidines. The common pyrimidines are the CMP, UMP and TMP. The de novo synthesis of purine nucleotides is somewhat different from the de novo synthesis of purine nucleotides. In case of pyrimidine nucleotides, the 6-membered pyrimidine ring is made first, and then it is attached to the ribose 5-phosphate, while in case of purine nucleotides, the ribose 5-phosphate is the starting material on which the purine ring is constructed. The process of pyrimidine biosynthesis starts with the formation of carbamoyl phosphate. This carbamoyl phosphate is also an intermediate in urea cycle for the synthesis of urea. The carbamoyl phosphate that is required in urea synthesis is made in mitochondria by the carbamoyl phosphate synthetase 1, while the carbamoyl phosphate that is required in pyrimidine biosynthesis is present in cytosol, and the enzyme is carbamoyl phosphate synthetase 2. Now this diagram shows the common ring, pyrimidine ring, and the sources of its atoms. This shaded area which covers the atom number 1, 4, 5 and 6. These atoms are donated by the aspartic acid, while the nitrogen at number 3 is derived from the glutamine, and the carbon at number 2 comes from the carbon dioxide. Thus, in the synthesis of pyrimidine nucleotide, the pyrimidine ring, the aspartic acid, glutamine and carbon dioxide are the sources of atoms. This reaction shows the synthesis of carbamoyl phosphate. The carbon dioxide in the form of bicarbonate ions and glutamine, they react with each other in the presence of ATP. Thus, the carbon dioxide and glutamine, their reaction is catalyzed by the enzyme carbamoyl phosphate synthetase 2, and the product produced is the carbamoyl phosphate. This reaction in which the carbamoyl phosphate is synthesized is completed in 2 steps. In first step, carbamate is formed, and then the carbamate is phosphorylated to carbamoyl phosphate. Now this diagram shows the reactions of the pyrimidine biosynthesis. In the first reaction, we see that the carbon dioxide and glutamine, they react in the presence of ATP to form the carbamoyl phosphate, and the reaction is catalyzed by the enzyme carbamoyl phosphate synthetase 1. Then this carbamoyl phosphate condenses with the aspartic acid and to form the carbamoyl aspartic acid, and this reaction is catalyzed by the enzyme aspartate transcarbamylase. Then in the next step, there is closure of the ring structure of the carbamoyl aspartic acid, and this closure of ring structure is catalyzed by the enzyme dihydroorotase, and this closure takes place with the removal of water molecule. So after the closure of this ring, the compound which results is the dihydroorotic acid. Then this dihydroorotic acid undergoes NAD dependent dehydrogenation by the enzyme dihydroorotate acid or dihydroorotate dehydrogenase. So after this dehydrogenation reaction, this dihydroorotic acid is converted to orotate or orotic acid, and this orotic acid is an important intermediate in the pathway of synthesis of pyrimidine nucleotides. Then this orotic acid is transferred to the ribose 5-phosphate, which is provided by the PRPP, and this reaction is catalyzed by the enzyme orotate phosphoribosyl transferase. And then after this reaction, the compound which is formed is the OMP, orotidilate. Then this OMP undergoes decarboxylation catalyzed by the enzyme orotidilic acid or orotidilate decarboxylase, also called the OMP decarboxylase. Thus, as a result of this decarboxylation, OMP is converted to UMP, uridine monophosphate. Then this UMP is phosphorylated to UDP and then to the UTP. Then from this UTP, the CTP is synthesized. For the synthesis of CTP, this UTP reacts with glutamine and receives its amide nitrogen. Thus, after receiving the amino group from the glutamine, this UTP is converted to CTP, and this reaction is catalyzed by the enzyme CTP synthetase, and this is an energy requiring process in which the ATP is utilized. So for the synthesis of CTP, ATP is required. Then UDP is the parent molecule for the synthesis of TMP. In the pathway of synthesis of TMP, thymidine monophosphate, first of all, the ribose sugar present in UDP is converted to its reduced form, that is the deoxy form. So this reduction is catalyzed by the enzyme ribonucleotide reductase, and the reducing equivalents are provided by the NADPH. Thus, the UDP is converted to DUDP. Then the phosphoryl group from the DUDP is hydrolyzed, and after the hydrolysis of this phosphate group, the UDP is converted to DUMP. Then this deoxyuridine monophosphate, it reacts with methylene tetrahydrofolate, which provides the methyl group to the DUMP. After the attachment of this methyl group to the DUMP, it is converted to the TMP, thymidine monophosphate, and this reaction is catalyzed by the enzyme thymidylate synthase. So in this way, the pyrimidine nucleotides UMP, CTPs, TMP, they are synthesized from the carbon dioxide, glutamine and aspartic acid, and orotic acid is the important intermediate in this pathway. Now these are the list of enzymes which catalyze the de novo synthesis of pyrimidine nucleotides, and this chart shows the difference between the CPS1 and CPS2. First difference is of their location, that is the CPS1 is present in mitochondria while CPS2 in cytosol. The CPS1 is an enzyme of urea cycle, while CPS2 is an enzyme of pyrimidine biosynthesis. In case of CPS1, the nitrogen is provided by the ammonia, while in case of CPS2, this nitrogen is provided by the glutamine. N-acetyl glutamate is the activator of CPS1, and PRPP is the activator of CPS2, while UTP is its inhibitor. So these are some differences between CPS1 and CPS2. Now this is the regulation of pyrimidine biosynthesis. The pyrimidine biosynthesis is regulated by the feedback inhibition at the 3 steps. The first step is the enzyme CPS2. The enzyme CPS2 is inhibited by UTP and is activated by PRPP. The second step of regulation of this pyrimidine biosynthesis is the aspartate transcarbamylase. This enzyme aspartate transcarbamylase is inhibited by CTP and is activated by the ATP. The third step of regulation is the orotidylate decarboxylase or OMP decarboxylase. This enzyme is inhibited by the UMP. During the synthesis of purine nucleotides, that is the CTP, we have seen that ATP is required for the formation of CTP, and the enzyme CTP synthetase is activated or stimulated by the GTP. So for the synthesis of CTP, both the purine nucleotides ATP and GTP are required, which ensures a balanced synthesis of purine and pyrimidine nucleotides. Thank you.